Okay, this is kind of the second part of the universal genetic code. So let's look at, okay, so here's a normal human, okay? And we know that we have DNA, and we know that this DNA, right, is made up of nucleotides, and the sequence of those nucleotides are your genes. So every 3 nitrogen bases, every 3 nucleotides is a gene. And let's say, for example, this is just one section. So we know this would go on for, 10,000 more bases. Okay, so just reminding us, this is a gene here, and obviously this is DNA. And during transcription, this would be converted into a long section of messenger RNA. So T would become A, C would become G, A would become U, and then GAT would be CUA. Okay, and we know that 3 nucleotides, 3 bases on messenger RNA is called a codon. Now, at translation, AGU, if you go read your codon chart, AGU codes for serine, and CUA codes for leucine. So that would be just two amino acids in a long chain of amino acids that make up a polypeptide, which then will fold up to become a protein. So for example, let's say that this one folds up to become the protein or the hormone insulin. Okay, so this sequence of DNA here, oops, that shouldn't be there. That sequence of DNA there codes for insulin. Okay, great. That's what normal healthy people do. They use their DNA to transcribe it into messenger RNA to make a sequence of mRNA, which they then translate into a sequence of amino acids which make up our proteins. Okay, what if you're diabetic and you either lack the sequence of DNA to make insulin, or you have a mistake in the sequence of DNA, so you don't produce insulin properly? All right, well, you may well have to take insulin shots on a daily basis to help control your blood sugar. Where does this insulin come from? Well, if you recall, when we talked about recognizing that the components of the genetic code have common organisms, right? What we're going to do is do this. We're going to take a bacteria, for example. Now we know that bacteria contains DNA. Okay? So here would be bacterial DNA. But what I'm going to do is this. I'm going to sneak this. Let me draw the bacterial DNA in red. What if I take this DNA and I cut it open? So I've now got a gap in there. And then I take this section of human DNA, which I can isolate, and I'm going to sneak it into here, like this. Okay? What I'm going to do is I'm going to splice human DNA into the bacterial DNA. Okay? Because the components that make up the genetic code are common to all organisms, the bacteria does not even realize, all right, this bacteria here does not even realize that it's got a section of DNA that doesn't belong to it. So when at translation you look at that DNA, right? Transcription, the DNA opens up. The sequence of DNA here is transcribed into the messenger RNA. It's exactly the same as if it had come straight from a human. And then you get translation, and that becomes a section of amino acids in a long chain that make up ultimately something that is genetically identical to human insulin. Right? It's called a human analog insulin. So what we can do is we can use bacteria, which are cheap and easy to grow, to make human insulin very quickly, which is what then can be purified and used by pharmaceutical companies to produce the insulin. This is the insulin that people who have to use insulin shots use. The only reason we can do that is because the components that make up the genetic code are identical to all organisms.